Hello, Professor. We are not able to hear you because it's muted.
They can hear.
Okay, for the students in the Zoom, um, we just started the individual the concepts on this topic development, and these are the objectives for today: fertilization, implantation, gastrulation, germ layers, inheritance, uh, germ cells versus somatic cells. How do we go from one single cell to multicellular organism? Why the cells are different? The difference between genotype and phenotype, and finally, the model organisms. We are going to start with fertilization. This is a very regulated process, and it's cyclic, and it's controlled by the brain. Starts with the hypothalamus that secrete or produces gonadotropin releasing hormone that will act on the pituitary gland or the hypophysis and is responsible for producing two peptides, two essential peptides, which are FSH, follicle stimulating hormone, and LH, luteinizing hormone. And FSH is responsible for acting at the ovarian follicle, which is responsible for generating estrogen. And then LH at some point will surge, like you observe here, and will lead to ovulation. After ovulation, the cells of the corpus luteum, which is corpus yellow, produce progesterone. So the ovary produces two main hormones, which are estrogen in the first half, like this, and progesterone in the second half, like this. Okay? It's not essential, it doesn't go all the way down, but estrogen plays a role in the first half of the cycle and progesterone in the second half. And LH is responsible, it's almost at the middle of this cycle, and is responsible for the ovulation. There is a surge and then there is ovulation, okay? And this is another view in which we have an immature follicle with the oocyte inside that needs to mature before it is released into the uterine tube or fallopian tube. So essentially, it gets mature and mature over the days, and when it reaches complete maturation, the oocyte is released from the ovary into the fallopian tubes. And then the rest of a period, half a period, post-ovulation, we have very important processes in the ovary, which is the production of progesterone, for example. The regression of this process, there's a lot of death of cells. There are lots of different regression processes, cell death, etc., etc. But importantly, this is the time of progesterone production. Okay, so what do the ovarian hormones, estrogen and progesterone too. Estrogen is the uterine wall with the endometrium, the lining of the uterus, and there are different processes. Estrogen is acting at the proliferative phase, makes the lining of a uterus or the endometrium to proliferate. That happens every month, okay? Every 28 days. So we have one stage here, and then when the oocyte is being released and progesterone is being made, we have sort of a maturation of the endometrium with the development of glands, some special and typical glandular formation, and the angiogenesis and other processes. So there are two main processes. What happens if the oocyte is not fertilized? What happens is that the level of progesterone declines here, and with that, the endometrium regresses, and finally, it gets all this tissue gets necrotic and eliminated. Okay, and this happens every month for many years. Okay? And then there is an ischemic phase, the endometrium that dies, and then menses begins. Okay, and it continues again with the next cycle, the continuation of menses, and then again the proliferative phase. So this is how it looks in the uterus. This is the ovary. We have the two phases, pre-ovulation and post-ovulation. As you can appreciate here, there's going to be a higher schematic to show you the different phases. But essentially, this is the maturation, and then the release of the oocyte, and this is the regression. Here, estrogen, here, progesterone, okay? This is a magnification. In an schematic, you have an early primary follicle that gets mature and grows in size. The oocyte is a giant cell. It's very big, okay? But the rest of the follicle needs to mature. And that is being done by estrogen. Okay? Estrogen is responsible for that. And once it matures, it is released. And interestingly, the ovary and the fallopian tube are not completely sealed. There is some space there. So essentially, the oocyte is released to the void, okay? And the fallopian tube is responsible. It has like protrusion fingers here, or it's like digits that go along with the oocyte that is being released and catches it. And then the oocyte is able to climb up into the fallopian tube. That's a very interesting process. So it's been caught by the digit-like structures here, which are called the fimbriae. All right. So each follicle contains a single oocyte. It's immature. They are periodically initiated to grow and develop, resulting in ovulation of usually a single competent oocyte in humans. This is another way to visualize an overlap between the hypophysis hormones and the ovarian hormones. So we have here the hypophysis or the pituitary hormones, which are the LH and FSH. The LH is responsible, luteinizing hormone. There is a peak, a surge, and that is a trigger for ovulation here 1 day after, and the preparation of a corpus luteum in the ovary. I'm repeating all these concepts because they are so important. Okay, and also the maturation at the of the follicle, and at the uterus level, you can appreciate here that we have estrogen playing a role during the proliferation stage in which the endometrium is growing and growing. And then it gets all the uterine glands and capillaries and vasculature. It's both phases are so important for the implantation of the embryo, and everything is coordinated, as you will see. Okay, so there is maturation of a sperm also, which is a very specialized cell, and that occurs at the epididymis during the first 2 weeks. Then the sperm cells, they travel and they are being added with seminal vesicule, fluid from the seminal vesicule, and fluid from the prostate. And all these components are essential for the sperm to be released, and also for survival in the maternal tract. Okay, so the sperm is deposited at the upper vagina, 10 to the 7 cells typically, and then it starts climbing up, and you will see that it has a tail that provides the sperm with motility, and the more it can swim, the more chances to be fertilized or to fertilize. So we have the sperm here, then it travels all the way up to the cervix, and you can appreciate that from 10 to the 7, we go down to 10 to the 6 at the cervix, and then 10 to the 5, 10 to the 4, it gets selected out, the sperm cell, depending on, for example, there is a rapid elevation of a pH. Not all the sperm cells survive, so we have one order of magnitude that disappears. And then through the passage of a uterus, some cannot climb up, and there are some contractile movements of the fallopian tubes and uterine tube to help the sperm keep climbing up and try to meet with the oocyte that is waiting at the upper portion of the fallopian tube. Okay, and in the end, we end up with 10 to the 2, which is 100 to 1,000. It's very, very small number, but yet it is more than expected. All you expect is 1, 2, or 3 sperm cells to fertilize, typically 1 sperm cell to fertilize, but we have in the range of 100 to 1,000. So there must be other selective mechanisms to say, you stay out, you can keep going. Okay, there are like checkpoints, and we see some of those. So this is the sperm cell is very specialized. It doesn't look like a eukaryotic cell. It looks very different. Every part is essential of the sperm. However, the most important part for fertilization purposes is the genetic component, which is in the nucleus. Okay, so when upon fertilization, there is a male and a female component that cross over and form the new genetic information. You are going to appreciate here lots of mitochondria. And now you know mitochondria equals energy. The sperm cell has to spend a lot of energy to swim up. Okay? Then we have the tail here very clearly. And then we have the acrosome. The acrosome is the cavity that provides with enzymes that will help the sperm get through a bunch of cells and get to the oocyte. We will see it in the next slide. Okay. So this is what it happens. We have the oocyte, the giant oocyte that I told you about, and we have layers that surround that oocyte. We have the space. It's called a perivitelline space. Here we have, importantly, what it's called the zona pellucida. This layer here that envelopes the oocyte. This is made by glycoproteins that play lots of different functions. One of those that it's for this moment is it prevents the oocyte from sticking in a place that is not that shouldn't stick. This is an ectopic place. Okay, if the oocyte has the zona pellucida, it will never stick at the fallopian tube. Okay, and that is good. But this zona at some point disappears, and when it disappears, the oocyte becomes sticky, and then it binds or it sticks to, and at that point, the embryo, the fertilized embryo, is at the uterus wall, and it can attach to the uterus, can implant to the uterus. So it helps the embryo to travel through the fallopian tube and meet at the uterus and then implant correctly. So that is one of the functions of the zona. It has several functions, very important. And it's also called the receptor of a species because different species have different zona pellucidas, and the sperm can bind only, has selectivity for just the zona of that same species. If you have a different species, then the sperm will not bind to the zona and will not fertilize the oocyte. So then we have here the cytoplasm of the oocyte. We have the genetic component arrested in meiosis 2, the second meiosis. And it's waiting, the genetic component is, or the chromosomes are waiting for the sperm to fertilize the oocyte, and then meiosis will be completed. Okay, so that's another checkpoint. Then there is a polar body, which is what remainings from the process of meiosis. Then we have cells here that surround the oocyte. Look at the difference in size. This is one cell, and the oocyte is one cell as well. That's why I'm saying that it's a giant cell, okay? And all these cells are cells that are dragged along with the oocyte when the oocyte releases from the follicle. These are follicular cells that remain attached to the oocyte, and it's called corona radiata. These are layers of follicular cells, okay? And this is the sperm that needs to get through the coronary radiata cells. That's what I was saying, that 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 the sperm needs to get rid of these cells and go through the cells and bind to the zona pellucida. And this is a magnification with the sperm here, with the acrosome fluid, or responsible for the acrosome reaction, or acrosome containing enzymes that will get through the coronary radiata cells. And then the sperm will find and bind to the zona pellucida. And that triggers the release of the nucleus into the oocyte here, the most important aspect of this fertilization, okay? There is a fusion involved between the membrane of the sperm and between the membrane of the sperm and between the oocyte. And then most of the components are being released. The most important one is the nucleus with the genetic information. Okay, so as I told you, 100 sperm cells or 1,000 sperm cells are too much, okay? It's much less than how it started, but it's still too much. So there is a need for other mechanisms of control of the sperm cells. And that is called prevention of polyspermy. Many sperm cells can bind to the oocyte, but only 1 or 2 or 3 can fertilize that oocyte. How does that happen? First, it needs to bind to the zona pellucida, okay? But all of them may be close to that. But as soon as one of them binds, there is a process, again, that it's called zonal reaction, in which the membrane of an oocyte suffers some alterations. One is an electrical change, and one is a chemical change. First, the electrical change that is short-lived, but it is immediate. So the sperm finds the zona pellucida, immediately the oocyte will suffer a change in the membrane, an electrical change that will prevent other sperm cells from binding and recognizing the zona. But that is short-lived. Then there is a chemical change, chemical reaction, and that involves cortical granules and calcium and the release of polysaccharides, etc., and enzymes as well that will eat up the zona pellucida, will degrade the zona pellucida. So that is long-lived, meaning that oocyte will never have another sperm bound to it, okay? Because the zona pellucida, or part of the zona pellucida, is being degraded. In particular, one component that is called ZP3, the receptor of the species. Okay, all the rest of the zona pellucida remains. Remember what I told you, that the fertilized egg or fertilized embryo needs to travel with the zona so that it doesn't implant ectopically. Okay, so this is a representation of what happens when one sperm binds to the zona. It binds to a zona. The first thing it happens is an electrical depolarization. That makes impossible for other sperm cells to bind. but short-lived, then it comes the long-lived process, which is the chemical depolarization, chemical changes that will secrete enzymes, degradative enzymes that will destroy part of the zona. Okay? Together, these two processes are called zonal reaction, and they limit the amount of sperm cells bound to the zona or fertilizing the oocyte. Okay, so far you had an oocyte on one hand and a sperm cell on another hand. But you never had an embryo. The embryo is defined as when both genetic components are mingled up. So once the sperm comes in and releases the nucleus and meiosis is completed, then we will have the embryo. Okay? It's a unicellular embryo. It's only one cell, right? It's not a oocyte because it has what was provided by the sperm, and it's not a sperm either. It is a new entity, the sperm. So this is how it looks. The egg, sorry, not the sperm, the egg. So we have the egg, which is the fertilized oocyte, with the two pronuclei here, one provided by the male, one provided by the female, and then remains of the meiosis, which are the polar bodies. Okay, two polar bodies, and the fertilization triggers rapidly what it's called cleavage. Cleavage is when one cell clips into two cells, all right? Meaning that there is a mitotic event, mitosis now, okay? But the cell doesn't grow in size. That is a special kind of process. So the DNA is separated, mitosis, but it doesn't grow in size. And you can appreciate this here. One cell, giant cell that is contained in the zona pellucida. This is the zona pellucida. This in green is the zona pellucida. And then the fertilized egg has divided mitotically, okay? And then from 2 cells, it goes into 4 cells, and then 8 cells, and 16 cells. And here we are at 16 cells, all right? There are several observations here. The morula, this is the stage of the morula. This is fertilized mouse egg, because this has been studied with the mouse a lot. Then we have 2 cells, then we have 8 cells. This is morula. At this stage, if we grab one of these cells called the blastomere, and we do some experiments, put them into uterus of a surrogate, we could grow an entire embryo, because all the information and lack of changes allows for each one of these cells to create potentially a whole embryo. It'll be smaller, but it'll be a whole embryo. Then it goes into 16 cells, and you can observe that the size of each cell gets smaller and smaller. That's because a process of compaction. The cell needs to, the 4 cells, the 8 cells, they need to fit within the zona pellucida. And that's why the embryo compacts the cells. And then there is another process, which is segregation. It's the first time that you see two types of cells. You see the trophectoderm here, and you see the inner cell mass. Okay, these are different cell types, and they will play different roles. The compact process, you can appreciate here the lack of border zones between the cells. For example, here there are like 16 or 32 different types of cells, but you cannot really distinguish between these cells. See? But there's they undergo a process of segregation and compaction. Okay. So this is another representation, a thorough representation in which the oocyte matures here. It is being released into the fallopian tube. The oocyte is arrested in second meiosis. It gets fertilized by the sperm, one sperm, okay, because of all these checkpoints and processes of degradation. And then you complete second meiosis and you start cleavage, two cells, etc., etc. Then the zona pellucida gets degraded. It is supposed to be degraded, okay? This is a physiological and natural process. And when it gets degraded and you have a more mature pre-implantation embryo without the zona and with the inner cell mass and the trophectoderm, it will be allowed to implant in the endometrium that has been prepared for implantation because of progesterone, okay? So progesterone is responsible for the maturation of this lining, and no zona pellucida, and the embryo implants, okay, and implants in the right place. If there wasn't zona pellucida here, it would have implanted ectopically. Okay, so this is everything is synchronized, okay, and makes and has a plays a role and plays a role. Okay, so cleavage consists of repeated mitotic divisions of the zygote, resulting in increased cell number. In human, cleavage is a slow 6-day process as the zygote moves through the uterine tube. Each cell of a divided zygote is called a blastomere. And remember that the blastomere can generate the whole embryo, one blastomere. And we will see next class that the blastomere is defined as totipotent. It could generate everything, okay? And all these blastomeres are identical at the morula stage. So as the morula migrates through the uterine tube, uterine fluid moves into the morula, creating a cavity. So the cavity that we see in the blastocyst here, the next stage, is made mainly by uterine fluid. Okay, so there is a role of the uterus, of the uterus horn here. Okay, this is a blastocyst. Once the fluid comes in, this is the blastocyst cavity. It's a cavity, okay? And these cells, there are cells that are more internal than the circle cells. So these cells are the external cells, and it's called trophectoderm or trophoblastic cells. Everything has 2 or 3 names, okay? Sometimes I say trophectoderm, sometimes I say trophoblast cells, but it's the same. And these inner cells, they are the inner cell mass. They conform the inner cell mass. Why is this important? Because the inner cell mass will give rise to the embryo. Okay? Only these inner cells will give rise to the embryo. The rest of the cells will play a role in invasion or implantation, will play a role in the generation of some of its layers, the amnion, the amniotic cavity, and the blastocyst cavity, etc., etc. But they will not be part of the embryo. It's called the extraembryonic tissues or extraembryonic layers. Another, for example, another tissue that is extraembryonic. Can you think of something else? Placenta, good. The placenta is an extraembryonic tissue, is made out of trophectoderm cells, okay? But not the inner cell mass. There might be inner cell mass components, but mainly trophectoderm. So this is how it looks, an early blastocyst. There are two sub-stages. Early blastocyst, here with the zona, and late blastocyst without the zona. This is the one that will implant. Okay, it's a sticky. This one is a sticky, the late blastocyst. This is another view or another schematic with all the processes, the maturation of the oocyte, the release of the oocyte upon LH surge, okay? The swimming of sperm cells, the fertilization of only one cell, the rest of the sperm cells die because they can't fertilize, and the finalization of the meiosis from second meiotic stage, it gets completed, and then we start mitosis, okay? And this is the process of mitosis. 2 cells, 4 cells, 8 cells, etc., etc. Then we lose, or the embryo loses the zona and gets implanted. And since progesterone was there, then the lining of the endometrium is well prepared for implantation. Okay, this is another view in which you can appreciate here all the glands and different formations are being present at the endometrium for implantation. It's waiting for the embryo to implant, and there are lots of different cell types and formations, like, for example, spiral artery, lacuna, capillaries, uterine glands, etc., etc. Okay. This is the process of implantation. Again, you can see that there is the epiblast, the cytotrophoblast, and the hypoblast. The inner cell mass develops into 2 different layers, into the epiblast here in blue and the hypoblast here at the border, okay? 2 layers, right? And that the blastocyst starts the process of implantation and mingling up with the lacuna and the different glands. So there is a mix of different formations here. And here there is more invasion. You can appreciate here that the embryo has invaded the whole endometrium, okay? And there is a mix of capillaries, etc., etc. The uterine gland, the embryo proper, the blastocyst, the primary yolk sac, etc. So it's a complex process in which both the maternal and paternal components intermingle, okay? And this is a graph, a schematic showing here the process of invasion with the trophoblast. Everything in embryogenesis is one layer gives rise to two layers, to four layers, etc., etc. So here we have the trophoblast now became cytotrophoblast and syncytiotrophoblast. The syncytiotrophoblast is made out of tetraploid cells, like meaning that there are 4 nuclei in these cells. It doesn't really matter, but these are essential for implantation. And then we have a cytotrophoblast also, very important. And here, the inner cell mass that gave rise to the epiblast, the blue cells here, and the hypoblast here. So these are the famous 2 layers that will compose the embryo at this time, 2 layers. But during development, we all have 3 layers, 3 germ layers, which is ectoderm, mesoderm, and endoderm. So what we have here is ectoderm and endoderm. Mesoderm we don't have yet, and we will see when the mesoderm kicks in, okay? So far during implantation, 2 layers, ectoderm and endoderm. In fact, this endoderm is not definitive, but it doesn't matter. It's endodermal in because of the markers that they produce. So this is one stage further in the invasion of the embryo into the endometrium. We can appreciate new cavities that are being formed, amniotic cavity, for example, the yolk sac, etc., the placenta, all made by the trophoblastic cells, because all these are extra embryonic tissues. The embryo is being formed by the epiblast and the hypoblast. Okay? And then at this stage, and then we'll see what happens later. So for the next stage, we use, just to visualize, we use the chick embryo. Why the chick and not the mouse or not the human? And the answer is because at this stage, they are interchangeable. They are very, very similar. In fact, many scientists use the chick to study the next process, which is gastrulation. And that is an important concept that you could pick any animal model to study human processes, because there are similarities, there are differences also, but there are similarities, okay? So this is the process of gastrulation. What happens here? The embryo attaches to the endometrium, starts forming the placenta and all the connections, okay? The umbilical cord, all that. That happens later. The very first stage is the formation of 3 layers: the ectoderm, endoderm, and mesoderm. And we see how it happens. And also the establishment of the 3 different axes of the embryo, that is also provided at gastrulation. The shape of the embryo, the elongated shape of the embryo is the first time you see an elongated shape of the embryo and axis of symmetry. So essentially what happens, there are a series of morphogenetic movements and processes in which these cells, the epiblast cells, these ones, the yellow ones here, they start moving into the center of the embryo, and they ingress what it's called the primitive streak. It's a streak that goes alongside our vertebrae here, our spine. okay? So this is what happens. There is a movement in and out underneath of the ectoderm. And when they egress or come out, those cells are mesodermal cells. They are converted into mesodermal cells. So at gastrulation, we have for the first time the three layers, the three germ layers. Ectoderm, which is this, and then mesoderm, which is formed by the ingression and egression of the cells, and then endoderm. The endoderm, these cells that I told you that they were not definitive, they are being replaced also by new endodermal cells that are created through this primitive streak, as you can see, right? So the most important concept here is that the three layers are being formed during gastrulation. And for this, you need implantation. And what is the primitive streak? The primitive streak, or where it goes along the spine. And it's like a zipper. It goes up first. The axis of symmetry are dorsal, ventral, proximal, distal, and forgot, proximal, distal. Oh, that is proximal, distal, and rostral caudal. Okay, so these are the three axes of symmetry of the embryo, and the primitive streak goes along the spine and it goes as a zipper. It goes from caudal to rostral, and then it regresses and goes from rostral to caudal, and then it disappears. So that's a structure that disappears during embryogenesis, okay, in the fetus or in the early embryo. That's the primitive streak. Along with the and this is another schematic. This is like a side view with when the primitive streak is about to disappear. This is rostrocaudal. You can also call it anterior posterior. So this is a side view in which you see anterior posterior portions and the end of a primitive streak. When it regresses as a zipper and goes down, it forms what it's called the neural tube, and we'll see more details about that. Okay, and there is also some names that you may want to remember: the Hensen's node, mesenchyme, the notochord. The notochord is another structure that appears during development, but then it disappears or regresses. In fact, if it doesn't regress, it means that it's not good, because it will form what it's called a teratoma, a very, very highly proliferative tumor. So the notochord has to disappear during in a normal process of development. Okay, there are many tissues or formations or structures that arise and then they regress during development. We will see that very often. And this is a ventral view. It's a view from above, okay, in which we have just the remains of a primitive streak and the neural tube here, or neural plate. These are different stages. And this is the prospective head, and this is the caudal portion. We don't have tail, right? But if we got a mouse, for example, we would have the tail here. Okay, so the primitive streak results from the proliferation and migration of the cells, cells of epiblast to the medium of an embryonic disc. Okay, so that's proliferation and migration of cells of the epiblast. the streak elongates by the addition of cells to its caudal end. That's the way it grows, okay? And this one view, this another view with the primitive groove in the primitive streak. There are many, many names. Most important one for you to retain is the primitive streak, this one, okay? These are epiblast cells, the ones that ingressed and then they egressed to form this void chamber here, and it's been occupied now by the mesodermal cells. There's another view, and I'm repeating different views because I appreciate that it's very difficult to visualize that. But perhaps at the end of seeing so many different views, you get an idea. In this view, you have here the primitive streak, and it's regressing. It went all the way up here, and then, so it did this. It did all these process here, like a zipper, and then it came down here. And this is the remains of the primitive streak. And at the tip of a primitive streak, we have what it's called the Hensen's node, which is a structure that is very important for signaling, for producing signals that will modify the environment. Okay. There's another view. This is the primitive streak. This is the Hensen's node. This is the epiblast in blue, and the cells, the epiblast or the ectoderm gets in and below the or ingress and egress and form the mesoderm. And then we have the ectoderm, okay? All this is going to be the embryo, but all this is extraembryonic tissues. This is just to show you how important are the extraembryonic tissues during development. It's not that that is dispensable. If you have a defect in extraembryonic tissues, the embryo probably will not develop to term. Okay? So it's essential. And there's another view in which you can appreciate the primitive streak that is regressing, and yet you have the mesodermal cells that come underneath and occupy all this void or empty space and will form the mesenchymal cells or the mesoderm that can be interchanged one name to another. Okay. So this is another view of the same, of the gastrulation, but now we are combining. We have the primitive streak, we have the neural tube, and at the same time, the embryo is growing, is proliferating. And that's why you start with this, but you end up the process of gastrulation with a much larger embryo, okay? And so here's the primitive streak that starts going up like this. And then you have next to it the neural plate, which will form the spine, okay? This is the neural plate. And you will have also the notochordal process, which is observed here at the dorsal portion, okay? So that's the notochord. This is the notochord again. Here, notochord, the neural plate, and we are in, or the neural fold here to form the spine, and that ends the process of gastrulation, okay? Okay, so now gastrulation, we have a trilaminar embryo. You remember that from two germ layers, we end up in 3 germ layers: ectoderm, mesoderm, and endoderm. Okay? And the mesoderm starts developing into something else. So not just mesodermal cells. They develop into somites, for example, which will give rise to, for example, dermis or myotome or sclerotome. And those formations will give rise, dermis is dermis, but the myotome will give rise to muscle, and the sclerotome will give rise to ribs. Right? So one structure will give rise to another 2 or 3 structures. That's the main concept of embryogenesis. And there is a process of neural fold here, and then it recircularizes neural groove here, and will end up with the neural tube. And that is a spine. Okay? So that's how the spine is being formed. And next to the spine, you have the vertebrae, which is mesodermal, and then you have the endoderm. So we have ectoderm, this, plus the neural tube, mesoderm in red, and endoderm in yellow. And then this keeps giving rise to more and more structures, okay? But that's the principle of embryogenesis. One layer to give rise to another layer, etc. Okay, so this is just to show you how colleagues or scientists can study the process of gastrulation. This is in the chick. Again, we could use a chick because the processes are very comparable. And this is to demarcate the notochord, for example. The notochord, something that I forgot to show you, is just, it's at the midline and just underneath the ectoderm. You see, in between the ectoderm and the endoderm. This is the notochord. It's like a rod-like structure that goes along with the embryo. Okay, here we see a transverse section of a notochord. Here, here, here. And it's very important because it is a machine that produces lots of different signals that are responsible for giving fate to the adjacent tissue. For example, it is responsible for determining that these cells that are next to the notochord will become motor neurons as opposed to the rest of the neurons. Okay? So it is very, very important. And when it does its job, it disappears. It regresses. Okay, we'll go back to this. This is just to show you the notochord, how it's been labeled here. And that's a typical way to study. And you can appreciate these like balls here that are next to the notochord. These balls are some evidence that we are segmented. We know that we are segmented, but a Drosophila, a fly, for example, it's much more obvious that it is segmented, that it has segmentation. We are segmented. We have the vertebrae, we have different, we have an evidence of the segmentation. The somites that are here, the ball of cells here, this is the first indication that we are segmented because everything is segmented here. Then all this undergoes morphogenesis and it will give rise to the muscle, which acquire different shapes. But originally, we are extremely segmented. Okay, the Hensen's node that I told you about, that it's at the tip of a primitive streak, which is essential for producing signals, important signals, is also responsible in part for the formation of an asymmetric kind of structure. For example, we know that we are very symmetric from outside. We have a dorsal, a ventral, we have axis of symmetry. But inside, we have one organ here, one organ there, the heart, the liver, the pancreas, etc. They are not strictly symmetrical. So somewhere has to the system had to produce some asymmetrical factors. Okay, so what happens, and this is the work of a very, very important scientist who looked at the Hensen's node very carefully and noticed that it had like little hair that it's called cilia at the top of a membrane, at the top of a cell, all right? And these cilia, for the most part, were inclined, tilted, okay? And it was tilted and it rotated during development. That provided or generated a gradient that goes from right to left or from left to right. And some of the factors that were produced are hanging in the fluid, they migrated from right to left, from left to right. And those are the factors that set up the axis of asymmetry. Okay, so we have symmetry, that's the most important aspect, but also we have asymmetry, and that was provided by the Hensen's node. Okay, there may be other systems around. The thing is that they haven't been discovered yet, but the Hensen's node is a clear example of right to left or left to right. For example, some of these molecular factors are lefty, lefty1, lefty2, nodal. These are all factors that are responsible for the asymmetry of this system. Okay, so here you can appreciate, for example, you can compare vertebrates and insects, and the two of them have the three layers. You could see, for example, here, ectoderm, mesoderm in red, and endoderm, the guts, etc., in yellow. You could appreciate this in vertebrates. Okay, this is a ventral view here, and this is a dorsal view. In insects, you see some subtleties, you see some differences. For example, the neural tube is ventral, but the organization is very similar. We have ectoderm outside, mesoderm in the middle, and endoderm inside, okay? So that is conserved between vertebrates and insects. So that's a good observation. And these are examples for ectoderm, skin, nervous system, for mesoderm, skeleton, muscle, kidney, etc., heart, blood. And for endoderm, the inner portions, the gut, the liver, the lungs. Okay? Then we have a concept of germ cells versus somatic cells. Here we have somatic cells. And if these cells undergo a mutation, the somatic cells will not be inherited into the progeny. So this is how these cells are being defined, somatic cells. Okay? There's no inheritance. However, if you have a germ cell, for example, the sperm and the oocyte, if there is a mutation there, imagine how important they are, because if you have a mutation and this can pass through progeny, right? And that's the principle of inheritance. A germ cell will allow passing a mutation into the progeny. And that's why they are very well protected in the body. Okay? Another principle that I would like to discuss with you is how do we go from one cell, how do we go into a multicellular organism? How come they are different? If they, for example, in identical twins, if the genetic information is identical, how come they are different? Okay, so this partly explains how this the organisms, the multicellular organism, is different, because there are different processes like cell proliferation, cell specialization, cell interaction with environment, for example, and cell movement. And all this may vary from embryo to embryo. And that is what it produces, a complete different multicellular organism. Yeah. So what it says here is the genome is normally identical in every cell. So every cell is identical. You have one cell that gives rise to 4 cells here, right? They're all identical, but then you have a process of specialization, different interactions, the interaction with the environment, for example, the influence with environment, cell movement, migration, etc. All this is responsible for producing a different multicellular organism. Another basic concept of embryogenesis is determination. And this is what embryologists did like 100 years ago, 200 years ago. They didn't have fancy equipment like microscopes or cell sorters or techniques more sophisticated like in situ hybridization. They didn't have all that. So what they did is they grafted different tissues and into different animals and studied what was happening. For example, the chicken whale kind of experiments in which chicken portions like this are being transplanted into other portions of the animal, of a quail, for example, which could be distinguishable easily or more easily than other systems, and then see what was happening. For example, in this particular experiment, one portion of a tissue of the leg of a chick, this is a chick quail transplant, but it was undifferentiated. These were all mesenchymal cells, chest cells. It was transplanted into a particular portion of the wing bud. Originally, they don't develop wings. They develop buds that end up forming wings. And what this experiment produced is a chimeric kind of wing in which the first portion was blue, the quail's kind of wing, but the distal portion was coming from the chick, okay? And not just coming, but it had like nails and other formations. It developed into something in which the origin was the leg bud. So that was a very, very typical kind of experiment. And cells have remembered positional values that reflect their location in the body. For example, here, these cells, they were undifferentiated at this time, but they had all the Q values in which if you transplant this, you will not become a wing, but you will become part of the, what it was before, which is the leg, okay? So that's why here you end up with a leg portion, despite the fact that you put in a bunch of cells, okay? So this is the principle of determination. The cells are already determined to become, for example, in this case, nails and toes, what nails, claws, toes, etc. Another process is induction. Induction is very typical in development. All these processes are very typical. That's why I'm enunciating. So in induction, you have, for example, the notochord. Just to mention one inductive tissue that will secrete factors that go right and left and will induce, these are inductive signals, and will produce different cell types here because of the environment, because of these producer cells, okay? So inductive signals can create orderly differences between initially identical cells. How do two cells with the same genome but separated in space come to be different? The most straightforward way to make cells different is by exposing them to different environments. In this case, these cells are being exposed to the notochord or to these central cell lines, okay? And induction may come in different flavors. For example, cell-to-cell contact here for short-range influence, or diffusion or endocrine factors, for example, that travel all the way from one tip to the bottom. And these are the long-range factors. This is another typical experiment that people remember very well. It was done by a scientist like 200 years ago. I don't exactly remember the date, Spayman, and it was done with a frog. And again, I want to emphasize the importance of dealing with different animal models for various reasons. In this case, the Spayman, what he did is he took a portion of a blastocyst of a frog. There is a blastocyst, but in the frog. And Spayman knew that this portion was very important. It was a source of signals because he's been observing this for a long time. And what he did is he transplanted that and said, and wondered what it was doing in a different environment, and put it here. Instead of here, he put it here, okay? And with surprise, he noticed that it created a secondary axis, a duplication of the embryo. But the most intriguing surprise of this experiment is that this embryo was made out of these cells, the host cells, not the donor tissue, okay? It wasn't yellow like this, you see? So what it means, or what he concluded out of this experiment, it's a simple experiment, but important in conclusion. What he concluded is that this region is a highly inductive region that could create, not with these cells, but with the host cells, another embryo, okay? So that's a little bit about the process of induction. These are typical inductive signals, which is which describes the diffusion, direct contact, and gap junction. Different types of inductive signals. Here, they are cell to cell, and this, for example, these signals travel from a channel and connect two different cells, and will become an inductive signal because it will induce on this cell some processes that are in the second part of the of this schematic here. Okay, the induction could be via direct contact. Here we have a receptor, a ligand, and a signal that gives rise to second messengers. And then diffusion here. Diffusion, for example, at the axon terminal, the release of some vesicular aggregates or vesicular proteins that will diffuse into a neighboring cell. So these were all concepts that they were discovered 100 years ago or more than that. We know now that the influence between cell and cell can be provided by the presence at the molecular level, the presence of a ligand that binds to a receptor, that it's located at the cell membrane, that will trigger a signal, for example, the activation of a protein kinase that will phosphorylate the protein that will translocate into the nucleus and activate the expression of specific genes. So we all know that, but imagine yourselves a few years ago, many years ago, performing these experiments without the knowledge that we have currently. And these are typical signaling pathways, ligand families, receptor families that I won't ask you to retain or remember. But if you want to read them just for to get an idea, it always comes down to 3 or 4 different families, which is the BMPs, the FGF, the WINs, the EGF, etc., etc. So you can study many different kind of processes in the embryo, and you end up all the publications that we have about embryogenesis. Very few describe newly identified factors. In general, these are the old ones, and we'll see next week when I teach you about stem cells that again they started looking at 20 different types of factors, and they end up always with the same ones. Okay, so that's for you to for curiosity. That it's something that it attracted my attention always. Okay, a new concept also during embryogenesis is the presence of morphogens. Doesn't mean that it doesn't exist in the adult, but at a much less rate occurs in the adult. It is almost a typical factor of embryogenesis. What does it do? And again, we go back to the notochord. In the notochord, we have like a radiation of different or secretion of different factors, right? Like there's one that it's called SHH. You don't need to know that. It's Sonic Hedgehog. And what it does is it is released by the notochord. It turns out that Sonic Hedgehog has a moiety of cholesterol attached, covalently attached to SHH, to Sonic Hedgehog. That makes it a little bit less a little bit more hydrophobic. So it travels through the extracellular space, but not quite easily. So it generates a gradient because of that. It's a slow kind of effort, right? And it generates a gradient. So the gradient now becomes something essential for this molecule because the system recognizes the concentration of this protein. And it says less than this concentration, it's going to turn into blue. A little bit more of the concentration of this molecule will turn into white, and a little bit more will turn into red. So it acts as a threshold kind of molecule or protein. And that's the concept of morphogen. There is a secreted factor, a signaling protein, but also it it responds to concentration, or concentration varies. Okay, and that is an important aspect of the function of the morphogens. And this is a typical example, and it's called the French flag because of the colors. But again, for example, under this concentration, under this value, it will form red. Under this value, it will form white, and under this value, it will form blue. Okay, so that's a new concept. And again, once again, the notochord is a tissue that contains these morphogens, many morphogens. Okay, so to support the notion that how do we arrive at a different organism if the genetic composition between two twin brothers is the same, right? And the answer is the genetic information is the same, yes, but there is, and start counting, enhancer regions in different tissues, and that will set the difference between two identical genes. There's going to be a promoter region that could be different between cell and cell. There is a coding region. There is some processing, like alternative splicing at the RNA level. Okay, and then out of a nucleus, for example, there's going to be translation and modification, like post-translational modification, PTMs. Okay, and also we will have, where is it? MicroRNA. I don't see it, but that's an addition to this. MicroRNAs that will be different from cell to cell. So all this facilitates the process of variability in the embryo. Okay, that's very typical. For example, if you find 2 or 3 isoforms of a protein in the adult, in the embryo, you could have 10 of those, and they will be different. These fetal proteins, fetal isoforms, are different. Hemoglobin, for example, there is a fetal isoform that has much less affinity for oxygen, and that creates a problem, right? So between fetal and adults, there are lots of differences. And we arrived at identical twins. Identical twins, very similar, very similar. You can pinpoint it, okay? But they're not identical. And they have an identical genome. The DNA is identical. So how can this happen? Because of all these variabilities that I told you, okay? So they have an identical genotype. If you, for example, if you genotype the cell types, the blood, for example, you will end up with an identical genotype, but the phenotype is different. and it has to do with the intrinsic variability of the individual, but also the environment. The environment makes a difference in the phenotype. Okay? That's why there was this experiment by the NASA in which two twin brothers went to, one stayed at Earth and the other one went to the uh, uh, to the uh, uh, to the space, and then when they stayed 1 year separate, and then they came back and they run lots of different analysis. It turned out that the differences were very subtle, and apparently this was because 1 year is not too much in the lifetime of these people. I don't have a definitive answer to that, but the people are very intrigued by the environment. Okay. Okay, so we are almost done. We have 10, 15 minutes. And we tackle model organisms. The question is, why do we study the model organisms? Why do we do that? Okay. Why can't we study those, for example, in humans? And you have any idea? Why do we need model organisms? Because there would be an ethical concern of studying humans. For example, if I want to study the development of a ribs fetus, okay? So there's always some kind of ethical problems with researching with humans. That makes a lot of sense. That's why we have different models. Some models are good for this, but bad for that. So there are pros and cons. And we're going to go over some of the advantages and disadvantages. So first of all, this is a mouse skeleton. You could appreciate that how similar is a mouse skeleton from another skeleton, for example, human skeleton. It's not too different. That's why we study model organisms, because the conclusions that we gather with a mouse embryo, those could be translatable to humans. Okay, we are not doing it just to understand how the mouse works, but we want to know if that could be extrapolated to humans. So they cannot be studied in humans. They have a similar set of developmental stages, the ones that I enunciated. Similar processes. We all share fertilization, zygote, cleavage, morula, blastula, gastrula. We have so many processes in common. We have the 3 germ layers: ectoderm, mesoderm, endoderm. We can study the chick and make conclusions for the human, right? There are similar underlying molecular mechanisms, morphogens, there are signals, transcription factors, etc. Okay. Again, this is to show you how similar they are. Look at this. This at the bottom here. Very similar. We have a mouse here, we have a fish here. Very similar. So if one day I typically work with mice, okay? But one day I want to say mice are too expensive, or something happened with the mouse colony, I want to switch to the zebrafish. I can do that. Okay? It will take some time to reorganize the lab and yourself, but you can do it. Model organisms are interchangeable. Okay. So we are going to go briefly over these different model organisms, but I just want you to retain what it's been listed up here, okay? Not the cycles or anything else that it's in the slide, just the list, okay? So the first one to study is, or the first one to describe is the amphibians, Xenopus laevis, the frog, okay? The frog is being used as a model organism. And the reason is because the embryos or the eggs can grow in tap water, cannot make it easier than that, okay? It gives rise to fertilized eggs that can be easily obtained by adding chorionic gonadotroph, if you wish, okay? You can stimulate them to fertilize. They are very large. You don't even need a microscope to see them. 1-2 millimeters, okay? Very, very big. They cleave at 20-minute interval. That means that it's very fast. So in a day, you can have all the results that you wish, okay? Instead of waiting, let's say 9 months, okay? Or 1 month for a mouse. They are resistant to infections. That's why we can grow them in tap water. They're very resistant. They don't contract infections. They're useful for molecular analysis, like gene protein expression and knockdown, like small interfere RNA or morpholino-antisense. So this is molecular analysis, okay? They're very easy. And also they can be used for molecular and electrophysiological studies. Then we have the birds. The birds, the chick, for example, I showed you the in situ hybridization techniques to study the notochord. Which are here probably, where you could see the stain here, the blue kind of report here. No, this is a transparent chip. This is a stain in brown stain, okay? This is an experiment showing the areas that are, for example, influenced by the notochord. So they are similar in morphological complexity to mammals, similar, not identical, okay? For example, we know they have eggs, right? They're easier to obtain and observe. In fact, you could grow the eggs, the fertilized egg, you could grow it in incubators, right? At certain temperature. The day you want to do the experiment, you open a door, you remove the egg, you crack a hole in the egg, then you do the experiment that you want. For example, you do some genetic analysis, you incorporate the gene, whatever you want to do. Then you close the window with Scotch tape and you put it back in the incubator. And 2 days later, you see what is the outcome of the experiment. So that's so cool, okay? So that's the chick. So microsurgical manipulations, what I was telling you. Knockdown analysis, morphological analysis, molecular analysis. We knock down, we reduce the expression of some genes, for example. In ovoelectroporation is another cool technique in which you crack open the window and you put 2 electrodes at the cheeks or at the gastrulation stage of a chick on both sides, and you put in the DNA, the genetic information, and you apply an electrical pulse there, and this will induce electroporation, which is the incorporation of the DNA into the cells. For example, the neural tube or the somites, etc. And then again, you put it back in the incubator, scotch tape, 2 days later, and you see, for example, if you incorporated LAG-seed, which is a visualization marker, you could see which are the cells that incorporated the DNA. Okay, so it's very, very, very easy to do. However, you may not want to study, for example, respiration in the chick because the gases, the oxygen, don't come through the placenta, for example, because there's no placenta, right? It's not a mammal. It comes from the air that diffuses through the shell and goes to the embryo. So there are processes that you can study. For example, placental development. If I ask you, can we study placental development in the chick? What would be the answer? The answer is no, because there's no placenta, right? Okay. So, and the nutrients, unlike us or a mammal that comes from the placenta, in this case, it comes from the yolk. That's why the yolk of the chick is so big, because it has to contain enough nutrients for the entire development process. Okay, and we don't have so much yolk, we don't need it. All right, this is an example of a technology that I mentioned to you, the in situ hybridization, in which, for example, if you're studying a transcription factor called TBX5, TBOX5, or TBX4, X1, doesn't matter. You could use in situ hybridization that hybridizes the RNA. It's not the protein, it's the RNA. And it creates like a hybrid with your probe that is labeled blue and the RNA that it's in the chick. And then if it recognizes the RNA of a chick, you will see some blue, right? So now we know that TBX5 is a factor of the wing bud, and that TBX4 is a factor of the leg bud. Okay, and this is kind of the study that you could perform in animal models. Okay, where is the location? For example, you found a very important gene, and you say, well, where is it located? Where is it made? Okay, and this is the way, one way to study it. A mouse. A mouse is very popular, very well studied. The whole sequence is known and refined and everything. And there are repositories in which we can request, we can ask, for example, if I need to eliminate one gene to study what's the role of that gene or what's the requirement of that gene, I don't have to make it myself. I just go to a repository and purchase it. Okay, it's a little bit expensive, but that is it's something that you could do. So that's very important. Nowadays, or in the past, transgenic mice, knockouts, that would take like between 6 to 2 years to get these mice and then the shipment and this and that. Now we can do all this with CRISPR-Cas. We can edit the genome. We can eliminate the gene, but also we can create point mutations. We can be at that level, okay? You can make conditional knockouts. For example, in our lab, we make conditional knockouts in which we supplant or we replace a serine that can be phosphorylated. We change it by a glutamic acid. Glutamic acid has a negative charge and it's similar to, it creates something similar to phosphorylation, but all the time. This is unregulated. And then we can study what's the effect of this hyperphosphorylation in the whole gene. Something very, so we can go at the very, very limit of the details using CRISPR-Cas. they are close to human, so it's a mammal, at least closer than chicks. The only, not the only, one of the problems is that it develops inside the mother. So if you want to study the embryo, you have to sacrifice the mother. That's a setback of using mice for embryogenesis. Zebrafish, also very popular. It's very good for studying screening, for doing screening of certain compounds. For example, you want to study heart defects, and you have a library of different compounds, okay, already prepared. You can purchase the library, and then you put one embryo or a few embryos per well in a fish. Okay, so you have like a battery of embryos, which, by the way, is transparent and it's easy to observe. So you have those there, and then you add one compound per well. And then you say, I like this phenotype because it slows down the heartbeat. So I want this one. And I get that embryo, and I sort of, I'm able to perpetuate that embryo by crossing, by genetic crosses. And eventually I'll get what I want. Okay? Some of the mutations are called ENU, erythronitrosourea, in which, as I mentioned before, we can use to screen some libraries. And we can do, again, a lot of knockdown and transgenic analysis, use CRISPR-Cas, etc. For invertebrates, the Drosophila, there is a combination of genetic and microsurgical manipulation, wealth of genetic studies, studies of transcription factors, role of segmentation. We can do lots of things, and you can appreciate that there's a lot of segmentation here, okay? And we can study transcription factors. And here, we can do some stainings in the larva, and we can make conclusions about the larva. For example, if we see blue here, we can see defects in the mouth, for example, or red here, defects in the antenna, etc., etc. The last one, I think, is the nematode, C. elegans, or Sinorhabditis elegans, which is a worm. And you may wonder, what is a worm doing? What is it? How is it helping us in our biological process? And the fact is that, first of all, it is also transparent, like zebrafish. We can, and also the organization of the cells of the body is invariant. So you could see a defect, and you could know right away that effect is done by this mutation. It's very simple to observe, and has a small number of cells. It is suitable for genetic analysis, and it's easy to disrupt function by RNA interference. There was a scientist who studied the length of the worm, but he studied a factor that it's called IGF-1 particularly, right? And one might say, what's the purpose of studying a worm here, or the role of the IGF in the worm? Turns out that a worm lasts for 8 hours, let's say, but there was a mutant, so they added IGF-1, right, to that. And there was a mutant that instead of dying in 8 hours, it died in 14 hours. Just that, right? And one might say, yeah, but that is not interesting. And it is very interesting because you can study aging with the worm. You can add, for example, factors and see if the worm lives longer, like IGF-1. It turns out that IGF-1 is a factor of aging. All right, and I think yeah, that's it. I'll stop here. Okay, thank you very much, and I hope I get some questions by tomorrow morning.